Hello everyone, Assalamu alaikum. Welcome to the new lecture on medicinal chemistry. This is me Faraz Kurban Rajpar, and in this video lecture I am going to discuss with you about the medicinal chemistry of cephalosporin. At the end of this lecture you will be able to learn about some basic history of the cephalosporin, structure activity relationship of cephalosporin, and the various structural modification of the cephalosporin. Cephalosporin. Cephalosporin are basically the class of antibiotic that belongs to the beta-lactam antibiotic. Why beta-lactam antibiotic? Because they contain the beta-lactam ring in their chemical structure. Cephalosporin were basically obtained from the natural source that are the Cephalosporium species. Some of the cephalosporin are later synthesized inside the laboratory by the chemical synthesis or organic synthesis by the semi-synthetic technology. By the attachment of the different functional group or the side chain at the 7-amino-cephalosporinic acid. The 7-amino-cephalosporinic acid is the basic ring skeleton that is present in all of the cephalosporin chemical structure. Cephalosporin chemical structure was first discovered in the 1978. In comparison to the penicillin, the cephalosporin are basically derived from the 7-amino-cephalosporinic acid, while the penicillin have the chemical ring skeleton that is the 6-amino-penicillinic acid. So this is one of the basic structural difference between the penicillin and cephalosporin chemical structure. But both of them, like cephalosporin and penicillin, both contain the beta-lactam ring. The only difference is the second ring that is fused with the beta-lactam ring. Like in case of penicillin, we have the 5-member thiazolidine ring, but now over here in case of cephalosporin we have a 6-membered ring, that is the dihydrothiazine ring. So due to the presence of this different ring, the antibacterial spectra of cephalosporin is different from the penicillin, and they have the greater activity against the gram-negative species in comparison to the penicillin. The modification of 7-amino-cephalosporinic acid side chain will result in the development of some useful antibiotic. That's what we say the different agents of cephalosporin. Like we have the 4 different generation of cephalosporin, like generation 1, first generation, second, third, fourth. All of these are basically differing from one another by the presence of certain different functional group in the side chain. Like first of these agents of cephalosporin was discovered by the Lilly in 1964 was the cephalothin. So this first discovered was launched into the commercial market by the Lilly company in 1964. Later in the 1948, Italian scientist Giuseppe Brotzu was the first one who identified the biological source of cephalosporin. He identified that cephalosporin are basically can be obtained from the natural source, that is the Cephalosporin acremonium, in the sewage outfall of the Sardinian coast. Abraham and Newton were the one who purified and established the chemical structure for the cephalosporin C. This molecule is the first of the cephalosporin antibiotic family, and later on this was used for the different derivatives that are basically synthesized by the structural modification in its chemical structure. So this is the basic ring skeleton that is present in the cephalosporin, that is the cephum ring or the cephum. So this is we have already studied in our beta-lactam medicinal chemistry lecture that cephum is basically the ring nucleus in which this beta-lactam ring is fused with the 6-member dihydrothiazine ring. In case of cephum, we have the double bond at this position. And in case of cepham, we have no any double bond and all the bonds are saturated. So in case of cephalosporin, we will have the substitution at this side. So numbering will start from here 1, 2, 3, 4, 5, 6, and this will be numbered as a 7. As in case of penicillin, this position of beta lactam ring has the number 6, because in case of penicillin we have a 5-membered ring, that is the thiazolidine ring fused with the beta lactam ring. So now let's learn about the structure-activity relationship of cephalosporin. Before going into the detail of SAR of cephalosporin, first let's learn about the basic ring skeleton of the cephalosporin. All the cephalosporin contain this basic ring skeleton, that is basically the composition of the two different heterocyclic rings. One of which is this beta-lactam ring that is fused with the 6-membered heterocyclic ring. Beta-lactam ring, that is the 4-membered ring, that is the common structural feature of all the beta-lactam antibiotic. Clear? This ring in case of cephalosporin is fused with this 6-membered heterocyclic ring, that is the dihydrothiazine. Clear? And then you have to place the common structural features, or the what we say the common functional group at the different site. Like at the third position you have to place the acetoxymethyl group. This one is the acetoxymethyl group that is present at the third number position of the cephalosporin chemical ring structure. And at the 7th position we have the acyl amino side chain. This one is the amide linkage, because carbonyl group is directly bonded with the amino group. So this is the acyl amino side chain. This R will be the different side chain or the different functional group in case of the different cephalosporin derivative. And at the 4th position we have the carboxylic acid functional group. So these are the common structural feature of all of the cephalosporin. So for drawing the chemical structure of any of the cephalosporin, you have to keep in mind this ring skeleton. First of all, what you have to do, you have to draw this beta-lactam ring and then simply fused with it with the 6-membered heterocyclic dihydrothiazine ring, and then place the acetoxymethyl group at the 3rd position, and at 4th position place the carboxylic acid functional group, and at the 7th position place the acyl amino side chain. Clear? So now let's learn about the different structural modification at the various site of cephalosporin chemical structure. SAR of cephalosporin. So basically the cephalosporin are the beta-lactam antibiotic in which the 4-member beta-lactam ring is fused with the 6-member 1,3-dihydrothiazine ring. So what is the function of these two rings? Basically beta-lactam ring is responsible for the binding of beta-lactam antibiotic, or over here the cephalosporin, with the penicillin binding protein to cause the inhibition of the transpeptidase enzyme to produce its antibacterial activity. Clear? So basically beta-lactam ring is essential for the antibacterial activity. The next is the 1,3-dihydrothiazine, that is the 6-membered heterocyclic ring. This ring makes the cephalosporin more stable toward the gastric acid media and more stable toward, or what we say, resistance toward the beta-lactamase enzyme. Clear? So basically these natural cephalosporin have the less poor activity in comparison to the penicillin, but their beta-lactam ring is more stable towards the acids and beta-lactamase, means their antibacterial activity is a little bit lesser in comparison to the penicillin, whose antibacterial activity for the natural cephalosporin, but their resistance towards the gastric acid media and the beta-lactamase enzyme is more. Then there are the certain derivatives that are made by the synthetic modification of cephalosporin side chain and at the 3rd position in their ring structure. They are basically the semi-synthetic cephalosporin. They have the broader spectrum of activities, and that's why they are synthesized by the substitution of the different functional group at the two different sites in their chemical structure, that is the R1 and R2 site, referring to the position number 3rd and to the position number 7. So these semi-synthetic derivatives are basically produced in order to have the cephalosporin with the wide spectrum of activity and with greater acid stability, with increased pharmacokinetic properties, means to make their absorption greater and their broader spectrum of activity, and to decrease the allergic reaction that can be occurring in case of the other cephalosporin. So the example of the cephalosporin that are produced by the semi-synthetic derivative are the cephalothin, cephadroxyl, cefuroxime, and so on, the different generation which are belongs to the class of cephalosporin. Now let's discuss the SAR of cephalosporin by considering the basic ring skeleton, that is the 7-aminocephalosporinic acid. Cephalosporin C has been identified from the 7-aminocephalosporinic acid ring nucleus, and it has the acetoxymethyl group at the 3rd number position. So let's consider the position number 1. You may observe that at position number 1 sulfur is present. If we are going to cause the oxidation of sulfur, it will be converted into the sulfone, and that sulfone derivative will have the lesser antibacterial activity. Besides this, we can substitute this sulfur with oxygen to convert the cephalosporin into the more active antibacterial agents, and those derivatives will be known as the oxazepam. Oxazepam is a cephalosporin derivative that has the oxygen in place of sulfur at the position number 1, and it has the greater antibacterial activity. At position number 3, you may observe that this acetoxymethyl group present. It is a highly reactive group, and it is the site of the metabolic degradation for the cephalosporin. And due to this, the cephalosporin are the metabolically unstable. So that's why it is being substituted with the different functional group and the rings to convert the cephalosporin into the stable derivative. Besides this, you may observe at the position number 3rd and 4th double bond is present, that is responsible for the ceph ring nucleus, and this group is this double bond or unsaturation is essential for the antibacterial activity of the cephalosporin. At position number 4, you may observe that carboxylic acid functional group is present. This carboxylic acid functional group can be used for the conversion of cephalosporin into the different type of the product, that are the ester derivatives, to convert the cephalosporin into the more stable pharmacokinetic derivative. Besides this, at the 7th position, you may observe that this is the acyl amino side chain. At this position, we can substitute this R with the different type of rings and other functional group to convert the cephalosporin into the stable derivative. We will discuss those derivatives in our upcoming slides. Now let's discuss why C3 acetoxymethyl group is substituted. The C3 acetoxymethyl group is present into the cephalothin and the cefaprin. Cephalothin is one of the very earlier identified, or what we say discovered, cephalosporin. This C3 acetoxymethyl group is metabolically unstable because it is highly reactive, and it is being rapidly metabolized or hydrolyzed by the esterase enzyme. So due to the hydrolysis, it is converted into the alcohol derivative, which are less active. So due to this, they cannot produce their optimum antibacterial activity. So due to this desacetylation of the cephalosporin, which is the deacetylation occurring due to this cleavage of acetoxyl functional group by the esterase, so they are going to be converted into the inactive metabolite. So to avoid this, there must be a need for the synthesis of those derivatives of cephalosporin which are metabolically stable. So those derivatives can be synthesized by replacing this acetoxymethyl group with the other stable functional group. So now let's discuss how C3 substitution can be made for the conversion of cephalosporin into the orally active cephalosporin. So first of all, let's consider the reduction of the acetoxy functional group. If we are causing the reduction of acetoxyl group, it will be converted into the methyl. And we have the different cephalosporin, that is the methyl group at the C3rd position in place of acetoxyl, and they are orally active and they are acid stable. The example of the drugs are the cephalexin and the cephalosporin. They have the methyl group at the 3rd number position. Besides this, we can also substitute this C3 acetoxy functional group with the chlorine. Like the chlorine is present at the 3rd number position in the cephachlor. Cephachlor, you can also memorize it by the mnemonics that the chlor in its name is indicating that the chlorine is present at the C3rd position. So there is a data that not for conversion of the C3 methyl group for compatible for the oral formulation, we must have the hydrophilic functional group in their chemical structure. So that hydrophilic group is being substituted at the C7 position. Amino group is being added. The C7 amino group at the C7 position is making the C3 methyl cephalosporin more acid stable and the more orally active. So all the cephalosporin like cephalexin, cephalosporin and the cephachlor, which have the methyl or chlorine at the C3rd position, they must also have the amino group at the C7 position, which is the hydrophilic group, and this hydrophilic group is making them stable towards the acid hydrolysis, or making them stable against the acidic media. So that's why they can be used for the oral formulation. So these are the example of the two drugs in the chemical structure of the cephalexin and the cephachlor. You may observe that this is the cephalexin structure. In cephalexin we have the methyl group at the 3rd number position in place of what? Acetoxy functional group. Besides this, you may observe that in the C7 acyl amino side chain, over here you may observe that this is the carbonyl carbon, this is the amide functional group, and at this alpha carbon we have the amino functional group, which is the hydrophilic functional group, and this is making them more acid stable and more compatible for the oral formulation. Same as in case of the cephachlor. In cephachlor, you may observe that we have the chlorine in place of the acetoxy at the position number 3, and in the acyl amino side chain we have this hydrophilic amino functional group. So these two structures are only for your understanding, they are not included in your exam purpose. So now let's discuss the C3 substitution for conversion of cephalosporin to the metabolically stable drugs. Means to convert the cephalosporin to those derivatives that are resistant to be cleaved by the esterase enzyme or other acid hydrolysis. So like first of all, we can convert the cephalosporin to the metabolic stable derivative like by substituting the C3 acetoxy functional group with the 3-carbamoyl functional group. This one is the 3-carbamoyl functional group, and in the cephalosporin which have the 3-carbamoyl functional group are the cefoxitin and the cefuroxime. Cefuroxime and cefoxitin have the 3-carbamoyl functional group, and they are orally inactive but metabolically stable. Means they are resistant to be cleaved hydrolysis by the esterase enzyme, and they are used by the parental route. Besides this, we can also substitute the C3 acetoxy with the pyridine heterocyclic ring, and when the derivative in which the pyridine heterocyclic ring is present is the cephalosporin, and the cephalosporin has also metabolic stability in comparison to the cephalothin or the cefaprin, which are metabolically unstable derivative, which we have already discussed in our first slide. So there is also another substitution, like we can substitute the C3 acetoxy functional group with another heterocyclic ring, that is the thiol ring. thiol ring. When the thiol ring is present in the C3 position, then that derivative is the cefazolin, and the cefazolin has the more gram-negative activity. Beside the substitution at the C3 position thiol ring, the cefazolin also has the substitution at the C7 substitution. At the C7 site, it has the tetrazole ring. Clear? So these are the different substitutions that can be made for the conversion of the cephalosporin into the metabolically stable derivative. C7 substitution. We can substitute the C7 position acyl amino side chain of cephalosporin with a different type of heterocyclic ring, other functional group, and those derivatives will have the good pharmacokinetic properties, means they can be stable for the oral formulation, and they can have the good stability against the beta lactamase, means they have the resistance toward the beta lactamase enzyme due to the presence of certain bulky functional group, or what we say steric shield. Like if we are going to substitute this C7 alpha carbon or the alpha amino group with the methoxy functional group, then the oxyamino derivative of cephalosporin can be produced. The example of the oxyamino derivative is the cefuroxime. Besides cefuroxime, there are other oxyamino derivatives as well, but here I have quoted only one example. Clear? So cefuroxime have the furan heterocyclic ring at this C7 acyl amino side chain. Beside the furan ring, it has the oxyamino methoxy functional group, and this will act as a steric shield, and due to this it is resistant toward the beta lactamase enzyme. Besides this, we can also cause the acetylation of C7 acyl amino side chain, and those derivatives will have the good antibacterial activity against the gram-positive bacteria. So this is the chemical structure of cefuroxime. Cefuroxime is one of the beta-lactamase resistant cephalosporin. Why? Because in the 7-acyl amino side chain, it has this amino-acyl methoxy functional group, and this functional group is making them stable toward the beta-lactamase enzyme, and it creates the resistance because it acts as the steric shield. Beside this, it has this heterocyclic ring, that is the furan, and this furan is indicated in their name as well, that is the cefuroxime. Clear? This is the chemical structure of the cephalosporin. In the cephalosporin chemical structure, you may observe that at the C7 alpha position, the methoxy functional group is present, and it will act as a steric shield, and that's why it acts as a resistant toward the beta lactamase enzyme. And beside this, it has the thiol ring in its C7 position. So the common feature between the cefuroxime is the C and the cephalosporin is the presence of the carbonyl functional group at the C3 position. You may observe that in both of these carbonyl functional group is present is the C3 position. This is making them the C3, or what we say metabolically stable cephalosporin. So these chemical structure are only for your understanding, they are not included in your exam, but you have to keep in mind the concept behind these functional group, that why they are resistant against the beta lactamase. Now let's consider the C7 substitution that can be made for converting the cephalosporin into the orally active. We have already discussed in our previous slide that we can substitute the C7 acyl amino side chain by having the amino functional group that will act as the hydrophilic functional group, and now these kind of derivative will be stable toward acid hydrolysis and will be orally active. The example we have already quoted, like the cephalosporin. Clear? Beside this, we can also substitute the aromatic phenyl ring of the C7 acyl amino, like present in the cephalosporin or present in the cephachlor, the benzene or phenyl ring that is present. We can also substitute that ring with other derivative, and now that derivative will have the more gram-positive activity because of the increase in their lipophilicity value. Beside this, we can also substitute the C7 functional group or the C7 acyl amino side chain with the phenyl glycyl ring, and when phenyl glycyl type of functional group is added, the cephalosporin will have the more stability, and that will result into the orally active cephalosporin, like the example of the cefuroxyl and the cephadrine. They have the phenyl glycol ring at the C7 substitution. So this is the overall summary of the cephalosporin SAR. We can make the substitution at the three different sites like C3 substitution. Main purpose is to convert the cephalosporin into the metabolically stable derivative or those which have the good pharmacokinetic property. Purpose of C7 substitution is to convert them into those derivatives that are acid stable or which have the beta lactamase resistant and which have the broader spectrum of activity. And besides this, we can also make the product of cephalosporin by the substitution at the C4-carboxylic acid functional group. So the chemical structure or the example of the chemical structure given in this video lecture are only for your understanding. I will not ask you in your exam the chemical structure of cephalosporin, but you have to remember the reason of each and every functional group that is present in their chemical structure. I may ask you that how can you synthesize the orally active cephalosporin, or how can we make the cephalosporin resistant against the beta lactamase. So you have to mention then the involvement of the different functional group that are responsible for those activities. So this was all about the SAR and medicinal chemistry of the cephalosporin.